Okay, this video is going to be about CRISPR Cas9 and its usage as it relates to humans. CRISPR Cas9 is a DNA editing system. We'll go through some more text on it. I just wanted to let you see the picture of what it looks like to start out with. And we'll come back to this picture after we talk about it some more. So here's normal DNA. Here are the DNA bases interacting with each other. Adenine, thymine, guanine, cytosine. And the line in the back is just a phosphate and ribose backbone. And now here's what CRISPR-Cas9 is all about. Cas9 is an enzyme to cut DNA, and this is the guide RNA to select which DNA target sequence is going to be cut. The Cas9 is like little scissors that cut DNA. The guide RNA is what finds the target. Then once the DNA is cut, new DNA sequence can be inserted. So there's a book called Codebreaker. It's written by Isaacson. It's a biography of a lady named Jennifer Doudna. She's from University of California, Berkeley, PhD, and Doudna Charpentier. She's a PhD lady from France. They work together to determine a lot of what CRISPR-Cas9 actually does. So the word CRISPR is an acronym for Clustered Regularly Interspaced Palindrome Repeats. Big fancy word. What this really is a bacterial immune system to fight viruses. Cas9 is just, we showed the scissors to cut the DNA. And why does this matter? Because what happens is a virus attacks a bacteria and it injects its DNA. When it injects its DNA, a lot of times it'll kill the bacteria, but some bacteria survive and they make a copy of that viral DNA. They stick it into their own genome, and now they've got a record of it. So when they're attacked by a virus again, they immediately can recognize this DNA and cut it up so the virus can't hurt them. And then what was realized was humans could, working with this in a laboratory, use this CRISPR-Cas9 system to edit genes and insert segments of DNA that we want to insert in there. And that potentially could have good usages. It could potentially treat disease like sickle cell anemia, which is a simple single base shift in DNA. Potentially eventually treat fancier diseases. They're looking at it for in vitro fertilization to potentially edit human embryos. They think that maybe it'll be used to eradicate a pest like malaria. And that's sort of the hope of CRISPR-Cas9 DNA editing systems. But there's also a lot of fear. If you can eradicate a pest like malaria, maybe some groups of people will be considered pests, and this will somehow be used to eradicate them. It usually ends up being a lot more profitable to hurt people than to help them. Unfortunately, I'm sorry to say that, but in my years of experience, that's kind of true. So universities will get billions of dollars for research. They do. All the American universities get tons of money for research. How often do they ever come up with anything useful? Almost never. And that's because they're always looking for grant money to make a profit for some corporation. And actually helping individual people, there's not much money in it, okay? There's very little research done in nutrition and toxicology. Most diseases are caused by problems with nutrition and toxicology. In the last 100 years, we got insulin, L-Dopa, levothyroxine. What else have we got from the universities? Where's our magic pill? People are fatter and sicker than ever with obesity, fatty liver, all these dietary diseases and toxicology diseases. Anyways, the guide RNA is made complementary to the target RNA, so the target DNA. So whatever, let's say there was a mutation, you want to remove the mutation. You can now sequence DNA. You can then target the guide sequence, make it to fit. That means match the base pairs, so it'll bind to that. Once it binds to that, then the Cas9 is the enzyme. It's a complex with the guide RNA. It'll then cut the DNA in that segment. Then you can put in donor DNA that you want, let's say a desired correct gene to fix the mutation. And they're using this now. One of the things being experimented with is they'll take organs, let's say from different types of animals, and try to modify them to make them more similar to humans in terms of their DNA and potentially transplant them. Of course, there's the Chinese system of just harvest them from people unwillingly, and it's a lot cheaper. Now, I purchased one of those kits, do-it-yourself kits at home, and I'm currently trying to edit the genes for the controlling part of my wife's personality. I don't know. I don't know what's going to happen. They do sell do-it-yourself kits. So anyways, one more time just to take a quick look at it. Normal DNA with the base pairs complementary to each other, the red and the black here. And then here's the guide RNA. This is synthesized by the laboratory team. and it will then bind with this target DNA, and then this enzyme around it, the brown outline around it is called Cas9. It then's like a scissors will cut the target DNA right around the guide sequence guide. Now you've got double-stranded break in DNA with this open gap. You take your donor DNA and you insert it into the test tube, and it can then be incorporated into that segment, and now you've got edited DNA. There's been DNA editing around like for GMO foods, for decades, but this makes it much cheaper and easier. It's no longer a specialized, expensive laboratory thing that only a few experts could do. Now it's something that is widely applicable, and lots of people are able to quickly learn how to do this. So what do I predict? I think it's going to mostly be a bad thing. I think they always exaggerate the benefits of it. So we'll see.